Hello everyone. So in this new video we are going to talk about the general principle of electrophoresis and the general principle of Sds Page, which is sodium duodecyl sulfate polyacrylamide gel electrophoresis. And please like and subscribe the channel. So let's start with the principle of general electrophoresis. Electrophoresis is a general term that describes the migration and separation of charged particles ions under the influence of an electric field. So of course electrophoresis is a general term where there are two electrodes, a positive electrode and negative electrode, and they're put into an electrolytic solution, right? So this positive electrode, anode and cathode, and there is electrolyte among it, and you put some charged particles inside it. So separation would occur. The positively charged particle will come towards this, and the negatively charged particles would move towards the positive electrode. So the migration and separation of these charged particles under the influence of electric field is electrophoresis. An electrophoretic system consists of two electrodes of opposite charge, anode and cathode. This is the anode and this is the cathode. They are positive and negative charged electrodes connected by a conducting medium called electrolyte. Now the separation effect of the ionic particles result from differences in their velocity, which is product of particles mobility and the field strength. So all it is saying is that the velocity v of the particle is equals to the mobility, that depends on the mobility of the particle, and the electric field strength, which is denoted by E, right? So this is a formula. Now the mobility m of the ionic particle is determined by the particle size, shape, charge and the temperature during the separation. So this m or mobility of the particle depends on size, shape, charge of the particle, right? And if this mobility changes of the particle depending on size, shape and charge, so of course the velocity of the particle will also change depending on what is the charge or shape or size of the particles, right? So this is electrophoresis. Now here is a diagrammatic view of gel electrophoresis. So you can see that this is the voltmeter and this is providing voltage to the, let's say this is cathode and this is anode, okay, and they are connected to each other, and this is a gel in which the samples are run, and there are electrolytes provided on either side, and these are the wells where we put the samples in these blue boxes. These blue boxes are the wells, known as wells, where the samples are put. We provide the samples within these wells of the gel. These are wells, and we put the samples inside it through using pipette. So what happens that then we switch this voltmeter on and electric current flows and a voltage is there. So this is the gel, right? So electric current flows. So the charged particles, depending on either the size or charge or the shape, they start moving towards the anode, right? So now you see, after the gel has been run, one of the molecule has come here, the rest are in the upper side. This means that this is a smaller molecule, this is a smaller molecule, small, while these are larger. Why is that? Because for example this has moved a long distance compared to these, so those smaller molecules moves faster within the gel and the larger molecule stays behind. So the larger molecule is still here moving slowly, while the smaller molecule has moved fast and had reached air. So this is a small molecule compared to these. So this is an example and diagrammatic view of gel electrophoresis. Let's go to the next slide. Electrophoresis of protein. So we know the general principle. Now we're going to talk about the specific kinds of electrophoresis. So let's start by the proteins. So sodium duodecyl sulfate, which is Sds polyacrylamide gel electrophoresis, also known as Sds Page. This stands for sodium duodecyl sulfate polyacrylamide gel electrophoresis, is used for the electrophoresis of proteins. Sds Page. Let's see its general principle. So SGS polyacrylamide gel electrophoresis or SGS page is the most widely used method for analyzing protein mixture quantitatively. It is particularly useful for monitoring protein purification, and because the method is based on the separation of proteins according to size, it can also be used to determine the relative molecular mass of proteins. So SDS page is only used for analyzing proteins or separating proteins molecules, and that too depending on their size of the protein. So what SDS page does, SDS page separates protein depending on their size. It does not matter what kind of charge the protein molecule has, it will only separate based on the size. So what will happen if this is the gel we run, and these are the wells, wells, and you put the samples in it and you run under a anode and cathode. So these run, so you will find some bands are here and here and here, and some are here. So these are the smaller molecules which have moved a large distance, small molecules of protein, and these are the larger molecule of proteins. So depending on the sizes of the molecule, of the protein molecule, they are separated in the SDS page. Now how this method works. So samples to be run on SDS page are firstly boiled for 5 minutes in sample buffer containing beta mercaptoethanol and SDS. So before we start to run the samples in the gel, what we have to do is we have to take the sample protein and then boil it with beta mercaptoethanol and SDS. Beta mercaptoethanol and SDS. We put it into a beaker and then boil it for 5 minutes along the sample. So what it does, the mercaptoethanol reduces any disulfide bridges present that are holding together the protein tertiary structure. So beta mercaptoethanol actually denatures the protein. Beta mercaptoethanol denatures the protein, right? And the SDS binds strongly to and denatures the protein. SDS also does the same. If it binds strongly to the protein and provides a negative charge to the whole protein. Now each protein in the mixture therefore fully denatured by this treatment and opens into a rod shaped structure with a series of negatively charged SDS molecules along the polypeptide chain. So we have, what we do is we take the sample protein and we boil it with beta mercaptoethanol, which destroys all the disulfide bridges within the molecule and denatures it, right? While the SDS molecules bind strongly to the protein, denatures it, and provide a negatively charged ion to all of the protein, right? So protein becomes something, a rod like structure, and there is SDS bind to all parts, right? These are SDS, negatively charged molecules. So this is the protein. Something like this happens after the denaturation of protein. Let's see the figure. So this is the figure. You can see protein folded with positive and negative charges. This is the protein in their tertiary structure. This is the tertiary structure protein. They're folded and there are disulfide bonds among it. You see disulfide bonds. These are the disulfide bonds. These are the protein molecules. Now what happens? You boil it with 2 mercaptoethanol. So what happens, it cuts. You can see this figure, disulfide bonds are cut from the in between. Something like this is formed. The disulfide bonds are broken. These are the disulfide bonds, these are broken, right? And the protein is denatured. Then you put SDS page. Page, not is only SDS, sodium dutyl sulfate. You put into it, and SDS is negatively charged, right? This is the SDS molecule with negatively charged. So these come in between. So unfolded linear structure with negative charge proportional polypeptide chain. So now this is a molecule of protein. You can see it is no longer folded like this. It is now in a long linear chain rod like structure with SDS bind all around it, giving a negative charge to all the parts, right? And now we put it into the wells of the gel to be run. So this is the function of beta mercaptoethanol and SDS. Again another picture. So this is the folded protein, and you put SDS into it and boil it. So what happens? Negatively charged molecules, the protein becomes a rod shaped structure, is no longer folded because it is denatured rod shaped structure, the protein, and negatively charged molecules are now bound all around it in the form of SDS. Right. So what happens next? The electrophoresis in acrylamide gel is frequently referred as PAGE, being an abbreviation of polyacrylamide gel electrophoresis. Now, cross-linked polyacrylamide gels are formed from polymerization of acrylamide monomers in the presence of smaller amount of N,N-methylene bisacrylamide, normally referred as bisacrylamide. So, and bisacrylamide is essentially two acrylamide molecules linked by methylene group and is used as a cross-linking agent. Acrylamide monomers is polymerized in a head-to-tail fashion into long chains, and occasionally a bisacrylamide molecule is built into the growing chain. So what it is saying is simple, that we already know what the function of SDS is. Now we have to know how the polyacrylamide gel is formed. So what we do is acrylamide monomers bind together to form a polyacrylamide gel like cross-linked polymer. Alright, but for that we need acrylamide monomers and N,N-methylene bisacrylamide. That's all. So let's see that in the next slide here. How the polymerization of acrylamide occurs. So this is an acrylamide monomer, this is a methylene bisacrylamide structure, and we need TEMED, which is your tetramethylene acrylamide, right? So, and we need this persulfate radical. So what happens? This is the acrylamide bisacrylamide, they polymerize to form this polyacrylamide gel, acrylamide. So cross-linked polyacrylamide gels are formed from the polymerization of acrylamide monomer in the presence of small amount of N,N-methylene bisacrylamide, right? So bisacrylamide is the most frequently used cross-linking agent for polyacrylamide gel. This is the cross-linking agent, the bisacrylamide gels, and this is the structure of polyacrylamide gel. So I can show you in another figure. This is a N,N-methylene bisacrylamide cross-linking monomer, and this is the acrylamide monomers, right? Two acrylamide monomers, and we need ammonium persulfate and TIMID, tetramethylene diamine, and for the reaction, and here it is, a polyacrylamide gel has been formed. This is a polyacrylamide. Cross-linking had occurred through this N,N-methylene bisacrylamide, and this is a monomer of acrylamide, another monomer. Only this is a bisacrylamide, right? The green one. Let's go to the next slide. Now we already studied about why this is required. We know why this is required. This is for cross-linking. This is for the uses monomer to form a whole polymer. But why ammonium persulfate and TIMID is required in this reaction? Let's see. So the polymerization of acrylamide is an example of free radical catalysis. So TIMID and the persulfate radical are used as catalysis. They're free radical catalysis, and is initiated by addition of ammonium persulfate and the base N tetramethylene diamine, timid. Timid catalyzes the decomposition of the persulfate ion to give free radical, that is a molecule with unpaired electron. So the polymerization of acrylamide is an example of free radical catalysis. Free radical catalysis. Right. So for this catalysis, this type of catalysis, timid, which is tetramethylene diamine, timid, and persulfate is required. Ammonium persulfate and NNN tetramethylene diamine is required for this free radical catalysis. So how this catalysis polymerization occurs? Polymerization of acrylamide is initiated by the addition of ammonium persulfate and the base tetramethylene diamine, which is timid. So we need ammonium persulfate and tetramethylene diamine, timid. So timid catalyzes the decomposition of persulfate ion to give a free radical. So we add timid and something like this happens. A free radical is generated, which have a this dot like structure shows a unpaired electron, and these are very highly reactive structure, very reactive, right? So this is how the catalysis occurs. So what happens now? Polyacrylamide gels are made from polymerization of acrylamide and bisacrylamide, where acrylamide forms linear strands and cross-linking of the strand is brought by bisacrylamide. So acrylamide plus N-methylene bisacrylamide, and polymerization occurs by the ammonium persulfate and timid by generating a free radical. So this free radical catalysis occurs, so polyacrylamide is formed. Thank you for watching this video. In the next video, we're going to talk about the whole how the whole SDS page functions, and how we can make the SDS gels, and how it runs, and how proteins are separated. Please like and subscribe the channel.